Now it's my great pleasure to introduce our second speaker, Jon Markert. Jon did his undergrad at the University of Wisconsin in biochemistry. He then moved to University of Colorado for his PhD. There, under Caroline Luger, he focused on how ATP-dependent chromatin remodelers disassemble and reassemble nucleosomes, which was unconventional back then. In 2021, he joined Lucas Fernand Lab in Harvard Medical School, where he has been using cryo-EM to study large multi-subunits machineries involved in transcription as well as replication conflicts. He has won multiple awards for his research, including the Fred Goldberg Postdoc Fellowship and several Best Poster Awards in multiple conferences. And with that, let's welcome Jon to share his research about H3K36 trimethylation. Yeah.
Thanks, Guy. So yeah, I'm Jon. I'm a postdoc in Lucas Farnung's lab here at Harvard Medical School. And today I'm going to be presenting two recently published stories. The first one is focusing on how nucleosomes are retained during chromatin transcription. And the second story is really how do cells keep track of what nucleosomes have been transcribed through histone modifications. So as we're all aware, the DNA inside of our nucleus is tightly wrapped around histones to form nucleosomes. And these nucleosomes really regulate genome stability or maintain genome stability by regulating when and where DNA can be accessed. And therefore, this nucleosome really serves as a barrier to the DNA replication, repair, and transcription machineries. So more about transcription. As RNA polymerase II moves along a gene, it recruits multiple different transcription elongation factors that surround RNA polymerase and make it more efficient at transcription. However, as I earlier mentioned, the nucleosome, which is found throughout the gene, serves as an actual barrier to the transcription machinery, meaning that nucleosome needs to be disassembled in order for the transcription machinery to progress. But it is essential that those histones are retained immediately after transcription and therefore redeposited on the DNA. This is a very coordinated process, specifically done by the histone chaperone FACT, which both does nucleosome disassembly and reassembly during transcription. Now, right now, we have a really good understanding on how FACT is recruited to the nucleosome, what it looks like when it interacts with the histones, and how it reassembles the nucleosomes in the wake of transcription. But what we don't currently understand is what prevents those FACT-bound histones from really flying away, because we want those FACT-bound histones to be maintained close to the transcription machinery, and therefore something needs to tether these FACT-bound histones to the transcription machinery to help facilitate the redeposition of the histones. So this is where the story really begins. And to do this, we turn to a fully reconstituted system where we have a nucleosome. And on this nucleosome, we create a single-stranded DNA that serves as a recruitment to a complementary RNA sequence. And together, they serve as a platform for the recruitment of RNA polymerase II. We then mix in multiple of those transcription elongation factors that surround the polymerase to make it transcribe more efficiently. So there are quite a lot of different transcription elongation factors, and they're going to be in the reactions throughout this talk. But I'm going to ask that you only really pay attention to certain ones, and we'll go through that as the talk goes on. So right now, just remember this factor RTF1. It will be colored pink throughout the talk. And really all you have to remember right now about RTF1 is that it binds specifically to RNA polymerase II. We then allow this transcription machinery to transcribe into the nucleosome with the histone chaperone FACT. We purify the complex and then visualize with cryo-EM. And you're going to see a couple 3D reconstructions throughout this talk. I'll try to simplify it as best as I can. For the most part, we'll think about this as RNA polymerase II with its transcription elongation factors present. And if you're looking at my slides, you can see that the direction of transcription is going into this direction to the right, which means any DNA that you see in this orientation is DNA that has already been transcribed. And now looking a little bit closer at that already transcribed DNA, we can actually see FACT bound, and it is in an extremely early stage of reassembling these histones. And then right next to FACT is actually that RTF1 factor that I asked you to remember just a few seconds ago. Now we're going to zoom in on this area a little bit more from a slightly different angle and just see, once again, we can identify most of the factors present. We have FACT in this beige color with the reassembled histones, and then RTF1 in, nearby. When we tried to model this structure with previous determined structures, we could pretty much model the entire density. But what we kept finding was this extra density that was unassigned, that couldn't be explained by anything that had been studied before. All we could say about this density is that it was sitting in a central cavity of FACT. And now that we're in a world with AlphaFold, what I decided to do was take every single factor that I have in this reaction and mix it with FACT and do AlphaFold predictions. And excitingly, only one hit came back, and that hit turned out to be a helix of that RTF1 molecule. And what we find is that that RTF1 is actually connected to the core RTF1 factor that is bound to the polymerase by a long, flexible linker. And together, this came into a model where we suggested that as the transcription machinery transcribes into the nucleosome, it recruits FACT. And as it recruits FACT, FACT comes in and is able to disassemble these histones. And to prevent those FACT-bound histones from flying away, RTF1 catches the FACT-bound histones and facilitates the movement of them onto the upstream DNA. So now we've answered this first question, which is really how does the transcription machinery tether the FACT-bound histones to facilitate redeposition. But the question that we were next interested in was really how does the cell know what nucleosome has been transcribed? And this has important implications on what factors get recruited to the nucleosomes after transcription. And the way cells do this is by depositing this histone modification called H3K36 trimethylation. And so what we know about H3K36 trimethylation is it's found exclusively within gene bodies. It specifically associates with activated transcription and therefore transcription elongation. And the enzyme that deposits this modification is SETD2. And SETD2 has several functions. It's important for recruiting factors that regulate chromatin spacing, factors that prevent cryptic transcription, and even has direct roles in regulating mRNA splicing. Now, if we look a little bit closer at SETD2, it's a very disordered protein. At the center of it, it has this catalytic domain. Directly adjacent to that catalytic domain is an autoinhibition domain. And at the very C-terminus, we have this domain that is responsible for recruiting SETD2 to RNA polymerase. Now, if I take this SETD2 construct and I mix it with nucleosomes and try to see H3K36 trimethylation, I see absolutely no activity. However, if I then remove SETD2's autoinhibition domain, I'm able to rescue activity. It's pretty much just saying that this autoinhibition domain is doing exactly as its name implies. It prevents SETD2 from being active, as demonstrated nearly a decade ago. But that kind of begins to ask the question, how can SETD2 function in cells given that it has this autoinhibition domain present? And we can begin to understand how this may be by looking back in literature, because not only is transcription associated, or not only is SETD2 associated with transcription, but some cell-based studies have suggested that it relies on specific transcription elongation factors. So now I'm going to ask you to pay attention to this SBT6 factor, which is colored blue right here, because previous work has demonstrated mutations in just SBT6 leads to global loss in H3K36 trimethylation. And recently, the Fred Winston group demonstrated not only mutations in that SPD6 leads to global loss in H3K36 trimethylation, but the only way that they were able to rescue H3K36 trimethylation was by mutating SETI2's autoinhibition domain, suggesting that there might be some type of link relating SPT6 to SETI2's autoinhibition domain. But exactly how that is currently unclear. So that leaves me with these three critical elements that regulate SETI2's function that need to be further explored, which is that transcription elongation is required for SETI2. It has some type of dependency on SPT6, and in the grand scheme of the reaction, SETI2 needs to be relieved of its autoinhibition. So to answer these questions, I'm going to turn back to the reconstituted transcription system, but now also add in SETI2. And I have the ability to simultaneously monitor transcription elongation and K36 trimethylation. So the first question I wanted to know is what happens if I just have all these factors present? So I'm not going to allow it to transcribe. I just want to know what happens when they're all together. So we can see that there's no transcription happening on this RNA extension gel. And simultaneously, I see absolutely no H3K36 trimethylation. But then if I allow the polymerase to actually transcribe into the nucleosome as observed with RNA product over time, we now see a massive increase in H3K36 trimethylation, demonstrating that the actual act of transcription is required for SETI2 activation. To explore why that is the case, we turn back to cryo-EM, where I take the transcription machinery, transcribe it into a nucleosome with SETI2 present, purify the complex again, and once again visualize with cryo-EM. And just as before, we're left with RNA polymerase 2 with its various transcription elongation factors. And it is now in the process of transcribing into a downstream nucleosome. And on that downstream nucleosome, SETI2 is bound. Now, let's look a little bit closer at that SETI2 bound nucleosome as it explains why transcription is activating SETI2. And that quite simply is because as the transcription machinery goes into the nucleosome, it induces nucleosome unwrapping. And that nucleosome unwrapping turns out to be a prerequisite for SETI2 recruitment, because if we look at a fully intact nucleosome, or rather a nucleosome that has not been transcribed, the DNA at the nucleosomal entry site serves as a steric clash for SETI2 binding. And so now that gives us this model where as the transcription machinery transcribes into the nucleosome, it unwraps the DNA to then recruit SETI2 to the downstream nucleosome and methylate it. Bringing this back into those elements that regulate SETI2 function, we've now explained why or how transcription elongation is required for SETI2's activation, but we haven't explained anything about the dependency of SPD6 nor how SETI2 can be relieved of autoinhibition. To kind of get to those later points, we turn back to the cryo-EM data, because when we look at the cryo-EM data just near this SPD6 factor and our upstream DNA, we notice this extra density. So you don't need to be a cryo-EMist to understand that this extra density doesn't really mean much. It just kind of looks like noise. But as I processed this extra density further, I learned that it was actually a second nucleosome bound by SETI2. Now, the way that we set up these reactions is we take a certain amount of RNA polymerase 2, which is kind of expensive and very large. And so we really want to have that entire RNA polymerase covered for downstream purposes. So we throw in excess of the different transcription elongation factors, nucleosome, and SETI2. So it's not surprising that we just have a random nucleosome or random SETI2 in our reaction. But what is surprising is that this second nucleosome is positioned directly upstream of the DNA and right next to this SPD6, because this suggests that this could be representing what a true upstream nucleosome, or rather a nucleosome that's already been transcribed through, and further suggests, because this happened so sporadically, that it might mean that SETI2 does not prefer to work on this downstream nucleosome, but rather that SETD2 prefers to work on this upstream nucleosome. So we can actually ask this question biochemically by manipulating that co-transcriptional reaction, where now instead of just allowing the polymerase to transcribe, we can engineer the templates to stop RNA polymerase at very specific spots. And so just as before, if I don't allow transcription to occur, I get no H3K36 trimethylation. If I then allow the polymerase to actually go into the nucleosome, just as demonstrated before, I get an increase in H3K36 trimethylation. But if I then allow the polymerase to go all the way through the nucleosome, where now that nucleosome is upstream relative to the transcription machinery, I get an even greater increase in H3K36 trimethylation, at least biochemically confirming that the upstream nucleosome is the preferred substrate of SETD2. We then wanted to understand this further by once again turning to cryo-EM, where I have the transcription elongation machinery. Now a nucleosome is placed upstream relative to that transcription machinery, once again with SETD2 present, purify the complex, and then visualize with cryo-EM. And once again, we're left with RNA polymerase 2 with all those different transcription elongation factors. But now in the upstream position is an upstream nucleosome. And on that upstream nucleosome, we have SETD2 bound. And excitingly, SETD2 is actually binding directly to SBD6. So now let's look a little bit closer at this SETD2-SBD6 interaction. So quite simply, we're able to identify the residues that interact with each other. So we know exactly what factor or what part of SBD6 binds to SETD2 and what part of SETD2 binds to SBD6. We've confirmed that this is the true interacting interface by taking wild-type SBD6 and wild-type SETD2, mixing them together, and seeing a complex forms. But if I if I break, or if I mutate either that interface in SBD6 or in SETD2, I'm no longer able to get them to interact. Just quite simply saying, we know exactly what residues interact with each other. They serve a greater purpose than simply binding, because if I mutate that and look at K36 trimethylation, I get a significant reduction in activation of SETD2. So now, not only have we explained how transcription elongation is required, but also a dependency on SBD6, but we still haven't explained how SETD2 can be relieved of autoinhibition. And if you remember from a few slides back, I showed some previous work from Fred Winston's group that had a genetic connection between SBT6 and SETD2's autoinhibition domain, suggesting, especially now with our knowledge that SBD6 directly engages with SETD2, that maybe SETD2 is being regulated, or SETD2's autoinhibition domain is being regulated by SBD6. But if we look at the structural interface, at least from a two-dimensional image shown here, the interaction interface between SBT6 and SETD2 is very far away from SETD2's catalytic domain and autoinhibition domain. So it doesn't really explain at this state how SBT6 binding could actually regulate SETD2's autoinhibition. But it turns out right next to this SBT6 binding interface of SETD2 is actually a helix. And this helix actually in three-dimensional space wraps and folds into SETD2's autoinhibition domain. And so what should be far away from the SBT6 binding interface is actually bringing the SETD2's autoinhibition domain and catalytic domain quite close to SBT6. And that means as the transcription machinery transcribes through the nucleosome, it is able to position SETD2's catalytic domain in close proximity to this upstream nucleosome, where it can then methylate this upstream nucleosome. And so if this is true, then this helix should regulate this entire process, meaning if I remove that helix, we should have the reverse effect, where SETD2 can no longer become activated because the catalytic domain and the autoinhibition domain can no longer be brought into close proximity to this SBT6 binding interface. And it turns out that's exactly what happens. So simply removing that helix, which has completely strongly reduces H3K36 trimethylation. And so now we've not only explained how transcription elongation and SPD6 are required for SETD2, but we've also demonstrated how SETD2 can be relieved of autoinhibition. And I would just like to add that there have been some preprints that have come out recently from the Patrick Kramer and the Sakina Kiramazaka groups that have confirmed several of these results, and I would encourage you to also look at these studies as well. And lastly, I just want to end by saying that the writers of these histone modifications is just one piece of the puzzle. We've also been focusing on not just on how these modifications are deposited, but how those modifications recruit downstream factors to the nucleosomes, and then finally how those modifications get removed. So lastly, I would just like to thank my advisor, Lucas, and the rest of the Farnung lab. We have a great community of science here in Boston, so I'm extremely lucky to have people like Fred Winson, Steve Gurutowski, Seychelle Voss in their respective labs to discuss all this work with. The Harvard and MIT cryo-EM facilities, the funding, Sky, Jackson, and the entire Fragile Nucleosome team for letting me speak with you, and I'd be happy to take any questions.
Thank you, John, for a wonderful talk. You got a great journey on the H3K36 trimethylation. If you have any questions, please feel free to type it in Q&A box or raise your hands so we can unmute you. So the very first question you got is, did you use a dinucleosome instead of a mononucleosome in your SETD2 cryo-EM studies?
Nope. So this was all done with a single mononucleosome. So when we had that in that first data set that I showed, like that random nucleosome that was positioned quote unquote upstream, that's just a random nucleosome that's in the sample. Yeah.
So I have a question. So in your system, it's like K36 trimethylation. I don't think it's like a serine phosphorylation. So if you add serine phosphorylation to your reaction, does this change the structure at all, or?
Yeah, so I guess this is related to the serine 31 phosphorylation.
Yeah.
Yeah. So yeah, this has actually been studied quite well. So serine 31 phosphorylation makes direct contact with some residues on SETD2, which stabilize SETD2, and therefore makes it more likely to associate with the nucleosomes. So we of course don't have that serine phosphorylated in our reaction, but if it is phosphorylated, that helps stabilize SETD2 on the nucleosome, and therefore you get an increase in H3K36 trimethylation.
So the second question you get it's the RFT1 helix contacting the upstream nucleosome, the same histone interaction domain identified by the Karen Aunt lab.
So the HMD domain, if I'm understanding correctly, is what they're referring to. Unfortunately, I'm not entirely sure I understand the question. But if it's referring to the HMD domain, it's right next to that interaction surface. It's literally just a few residues away.
Interesting. I think the next question the finding of the RTF1 helix holding onto nucleosome is really cool. Have you tried looking at the nucleosome dynamics in live cells to see if mutating this helix does indeed cause free diffusion of nucleosomes after transcription?
So no, I definitely have not done that. That's a really exciting question, and I think it deserves further studies. I have not done that.
So thank you for the great talk. Have you looked at the H3K36 mono and the dimethylation in vitro transcription reactions?
Yeah, we actually have. The reviewer asked us this exact question. So for whatever reason, SETD2, once it methylates that first lysine, it just instantly turns it into trimethylation. This has actually been explored in previous studies. It's a slight mutation in SETD2's, one of its aromatic residues in its active site. So we can't actually monitor any real detectable mono or dimethylation in our assay. It, of course, has to deposit the mono and dimethylation, but our idea is once it deposits the first one, it just instantly puts the other two on.
I guess the dynamic is too fast to check these ones. Yeah. What
Yeah.
is interesting is all these like remodelers, like as we are doing neurobiology, it seems to be involved in a lot of like neuronal degenerative disease. Yeah. I think that's pretty much all the questions. That's thank you, both speakers, Rachel and John, for the great talk. And I'm sure if you have any questions, you can reach out to them by email. They will be very happy to answer your questions. Yeah, thank you all for coming.